Good day everyone, I am Candy Plaza and welcome to my vlog. So for today's video, I am going to tackle about cell types and cell structures. But before that, let's know first what is cell. Cell, from Latin word cilia, meaning small room, the basic structural, functional, and biological unit of all known living organisms. The cell is actually too small to see with the unaided eye. It is visible here in such details because it is being viewed with a powerful microscope. Cells are consist of three parts: the cell membrane, the nucleus, and between the two, the cytoplasm. Cell membrane. Every cell in the body is enclosed by a cell or plasma membrane. The cell membrane separates the material outside of the cell or extracellular from the material inside the cell or intracellular. It maintains the integrity of a cell and control passage of material into and out of the cell. All materials within a cell must have an access to a cell membrane or the cell's boundary for the needed exchange. The cell membrane is a double layer of phospholipid molecules. Proteins in the cell provide structural support, form channels for passage of materials, act as receptor sites, function as a carrier molecules, and provide identification markers. Next is the nucleus. The nucleus, formed by a nuclear membrane around the fluid nucleoplasm, the control center of the cell. The nucleus determines how the cell will function, as well as the basic structure of that cell. And the last one is the cytoplasm. The cytoplasm is the gel-like fluid inside the cell. It is the medium for chemical reaction. It provides a platform upon which other organelles can cooperate within the cell. Within the cytoplasm, materials move by diffusion, a physical process that can work only for short distances. Cells were discovered by Robert Hooke in 1665, who named them for their resemblance to cells inhabited by Christian monks in a monastery. Cell theory, first developed in 1839 by Matthias Jacob Slenden and Theodor Schwann, states that all organisms are composed of one or more cells, and that cells are the fundamental unit of structure and function in all living organisms, and that cells come from pre-existing cells. Prokaryotic is the simplest and most ancient types of cells. If you take a look at the prokaryotic cells and eukaryotic cells side by side, you could see the number of differences. To begin with, prokaryotic cells are a lot smaller than eukaryotic cells and have the simpler structure. But bear in mind that this simple structure are actually a good thing for prokaryotes because it allows them to reproduce very quickly and very effectively. If you can take a look inside of the prokaryotic cells, you will probably surprise how simple it is. For example, prokaryotic cells are locked in nucleus. In fact, the actual name gives a hint about its structure. Kari means nucleus. Pro means before. So basically, the word prokaryote means before nucleus. Inside of prokaryotic cell is an open unit with no compartments. There are no membrane-bound organelles. However, small structure ribosomes are scattered throughout the cytoplasm. The cell's DNA is located in the region of cytoplasm called nucleus region. The nucleus region is not the same thing as nucleus because it is not enclosed of the membrane. Prokaryotes have the cell wall around the plasma membrane. It also possessed one or more flagellum used for movement. Some prokaryotes have an extra layer outside their cell wall called a capsule, which protects the cell when it is engulfed by other organisms, assists in retaining moisture, and helps the cell adhere to surfaces and nutrients. Pili are hair-like structure on the surface of the cell that attach to their bacterial cell or surfaces. The composition of cell wall depends on whether the cell is an archaeon or bacterium, that basically it consists of a complex polysaccharides. Prokaryotes have a large array of characteristics that enable to withstand different conditions, environment, and resources. Some live in the absence of oxygen, some in extreme cold or hot temperatures, and some in the bottom of the ocean where their only resources is hot hydrogen sulfide. One of the things you need to know is the structure of animal and plant cells. So in animal cell, first we have the nucleus. Its function is to basically command and control of the cell. It tells the cell what to do, how to grow, what proteins to make, and what lipids to reproduce. In the nucleus, there is a long strand of DNA known as chromatin, and the DNA is the basically instruction man or the blueprint of the cell. It stores the basic genetic information of the cell. And within the DNA, you have the instruction on how to make proteins, how to make the individual organelles in the cell, and what they need to do. Inside of the nucleus, we have the nucleolus. The nucleolus creates ribosomal RNA to make ribosomes. Ribosomes creates ribosomal RNA and protein. The ways nucleus makes protein is that it sends mRNA or messenger RNA, which carries the instruction to the ribosomes to make a specific type of protein. The nucleus is surrounded by a membrane called nuclear envelope, and on this membrane, you'll see a holes on it which are known as the nuclear pores. Nuclear pores allows stuffs to go into and out of the cells. So the messenger RNA comes out of the nucleus through nuclear pores. Now, let's talk about the ER or endoplasmic reticulum. There are two types of ER, the rough ER and the smooth ER. The difference between the two is that the rough ER contains ribosomes, while the smooth ER does not contain any ribosomes. Rough ER involves in protein production. It is called rough because it is stabbed with ribosomes. However, it does produce lipids, cholesterols, and hormones. It also assists in detoxification, which means breakdowns toxins. Now let's go back to rough ER. So, once the rough ER, by means of ribosomes, once they make proteins, the proteins are enclosed in the vesicles, and those vesicles get transported to Golgi body. Now, the Golgi body receives the vesicles and it modifies the proteins that are in the vesicles. And the way it does by adding carbs, lipids, and proteins. And it can also fold the protein. It can give the proper shape because the function of protein is dependent to its shape. Once it modifies and processes the proteins, it exports the protein out of the cell. Now, let's talk about the other organelles, the mitochondria. Mitochondria has its own separate DNA. It's different from the DNA of the cell, and its job is to perform cellular respiration. In that process, it takes energy towards fats and carbohydrates and convert it into a molecule known as the ATP, or adenosine triphosphate. The next is the lysosome. Its purpose is to break down foods. Lysosome also contains digestive enzymes, and in white blood cells, it can also destroy pathogens. When white blood cells engulf pathogens in the process of phagocytosis, the lysosomes endow white blood cells, it can break down those pathogens or diseases causes agents into smallest stuffs that can be used or recycled by the cell. Next is the cytoplasm, basically the jelly-like fluid that is in the cell. All of the organelles are dissolved in cytoplasm. It contains solute like salts, electrolytes, and other stuffs like carbohydrates and lipids. Now let's talk about the cytoskeleton. The function of cytoskeleton is to maintain the shape of the cell. At the same time, it provides the structure support. The cytoskeleton consists of a network of 3 fibers, the first of which is the microtubules, the second is the microfilaments, and the third one is the intermediate filaments. Now, how can we identify them into the cell? The microtubules is the largest of the 3 fibers, the microfilaments is the smallest of the 3 fibers, and the intermediate filaments is somewhere between the microtubules and microfilaments. The microtubules are made up of proteins called tubulin, and the microfilaments are made up of proteins called actin. The microtubules' purpose is to provide structural support and transportation service to the cell. So, the vesicles can move from one part of the cell to another part by means of microtubules. The microfilaments are involved in elongation and contraction, so they assist the cell to move. The next is the centrioles. They are active during the cell division, and during that time, they form something known as mitotic spindle, which consists of microtubules. What it does is that during cell division, it falls apart. Chromosomes allows the cell to split into 2. The chromosomes are condensed version of chromatin, so they form during cell division. And that are the animal cell. Now let's talk about the plant cell. Even though they are similar, plant cell has some characteristics that are not found in animal cell. The first of which is the presence of a very large vacuole. In vacuole, it stores water and nutrients, and at the same time, it provides structure support to the cell. The other organelle that is found in the plant cell but not in the animal cell is the chloroplast. The chloroplast is green due to the pigment chlorophyll, which play a role to photosynthesis. Photosynthesis is the process by which plants take the sunlight, carbon dioxide, and water and produces glucose and oxygen gas. Another feature that presents in plant cell but not in animal cell is the presence of cell wall. The cell wall surrounds the plasma membrane of plant cells and provides tensile strength and protection against mechanical and osmotic stress. And that would be all. I hope you learned a lot from this video. Thanks for watching!